everybody, welcome back. So I'm Elisa, and we are talking about the circulatory system, specifically looking at the blood. And now we're going to continue on with some more red blood cell information. Okay, so we looked at this. So the red blood cell, or the erythro, meaning red, cyte, meaning cell, right, is biconcave, meaning like this. It has this like concave right here, right? And biconcave on the other side too. So this is two, right? And so we get this like kind of ring and no nucleus, no DNA, very few organelles. Okay, here it is. So it's like very small. And like here's a capillary. We talked about that. And then it has direct contact. All of its heme groups, all of its oxygen and carbon dioxide transferring proteins are going to have direct contact with tissues more effectively. And we're not going to waste this inner space that they can't really touch, right? They can't touch it. Um, So let's look at a few things. We have a few different blood types, like quite a few. Like we'll look at the canonical groups, like the A, B, O groups, but just note that there's more to it than that. There are a lot of what we call surface proteins, and like surface proteins would be, there's these little proteins on the top of all these cells, and they're what cells use to communicate to each other, to stick to other things, to activate functions. And so some of those glycoproteins are just different. There's a heterogeneous human population for that. Okay, so glyco meaning like sugar, and proteins meaning protein also, glyco, sugar, and lipids meaning fats, right? So those are all some different surface molecules. So also the cytoskeletal proteins are kind of interesting. So actin is always going to be in there. Actin is a really important, the bones of most cells. And so most structural and also motion, right? So cells move their organelles around, they move parts of theirs, change their cell shape around is all heavily dependent on actin. And then spectrin usually appears as kind of a stabilizing protein. You can oftentimes see the spectrin on the surface of some of these cells, like the tetramer of spectrin. And so they give structure to the membrane, especially. They're kind of like a chain link fence that sits right around the membrane. And so they're going to give it that durability and that resistance because they are, running into other cells all day long, right? And as you can see here, they have to be pretty tough, pretty tough. There is stretchability, extensibility in red blood cells. They bend and squeeze through these small capillaries, as you can see here. Here's a capillary, but some are smaller. Like we're talking one RBC at a time, folks. Yeah, looking pretty small. Their key functions are not isolated, but remain primarily within gas transport. And it's not the exclusive mechanism of gas transport in the body. Primarily, they're doing O2 oxygen, right? That's going in to be used in metabolism, and then carbon dioxide, which is an end product of that metabolism. So this is a waste product typically going through. So because of that biconcave shape that we looked at, they have a really high surface area ratio. And so it means that they are going to make the most contact with the adjacent cells or the tissues they're trying to bring oxygen to or to take away those wastes from. So some of the loss of, due to the loss of the organelles as they mature, because remember they eject like the nucleus and they eject a few other elements, we have increased diffusion rates of certain substances. So other things are those cytoplasm is going to be packed with different stuff than other cells because they're really just about moving oxygen and carbon dioxide. So they have a really high percentage of this really cool molecule, which we'll look at, called hemoglobin. Let's see, here it is. So hemoglobin is, right here is this little iron, right? If you remember iron. Oh, here it is. There's a little iron, right? Here's a little iron. Is a positive, it's a cation, and it's what things like oxygen stick to. Oxygen just loves to stick to iron. And so this is one of the transport mechanisms. And it's as you can see, there's these four, subdomains or hemoglobin is made of these four components all kind of globbed together, right? And so each one of these is going to be like, popped together. so you can see. So we have kind of a clover leaf shape. Okay. And so that's the protein that carries hemoglobin, right? It carries that oxygen around primarily. It has a higher affinity for oxygen than it does carbon dioxide too. And so that's one of the reasons it helps kind of kick off the carbon dioxide, or I'm sorry, kick in the oxygen right into the seat, locks it in really well. So there are approximately 280 million hemoglobin molecules in only a single red blood cell. What? That's like so much because it can deliver a lot of oxygen that way, right? And it also helps transport carbon dioxide to the lungs, but it's just like really meant for oxygen. It's really meant to be an oxygen seat. It also has enzymes such as carbonic anhydrase or CAH in the cytoplasm. It produces carbonic acid from the CO2 and water, which is primarily the function of this enzyme. And so what that does is it allows us to transport degraded carbon molecules a little bit easier, and it also helps to buffer the pH. So all of the hemoglobins, as we looked at, remember we have the clover leaf conformation, right? Well, here's a little stem for fun, right? Of the globins. Some people also, one way to think of it is globs, right? Technically, they are kind of protein globs, globbed on there. Um, so an adult hemoglobin protein is going to have two alpha chains and two beta chains, and fetal is going to have two alpha chains and two gamma chains, right? And that has to do with the fact that, um, during pregnancy, the fetus is going to need to take oxygen at a higher efficiency, right, out of the mother's blood. So that's kind of really where that comes from. We'll talk about that a little bit more on. So we have the four heme groups. So they are a non-protein moiety, and oops, iron. Oh, I made a terrible. Can I erase that? Nope, not gonna let me erase that. Sorry. Sorry, everybody. And then iron, remember, is going to be the seat that oxygen likes to sit on, and there's four binding sites right in the middle of each one of these leaves for that iron. There's that structure of hemoglobin again, looking good, looking good. This is an adult one, right? Alpha beta chains. So, the efficiency of the erythrocyte in carrying oxygen around is going to be directly related to how many red blood cells are there. So this is when we look at the hematocrit. Remember, that's that packed cell volume, right? That little tube of blood that we took, and then we centrifuged it down, and we were able to look at the relative percentages. So generally, we sit at around, 50 to 50, just barely plus percent of RBCs in an individual. Um, Hemoglobin concentration of the whole blood after we look at it is still going to be pretty huge, right? So like 16 to 18 grams per deciliter. So that's, quite a bit on the high side. And the RBC count within that percentage volume, boop, is going to be sitting around that like 5.5 to 6 million per microliter. So tiny red blood cells. Okay, so they're very, very, very small cells. Um, so the values in most of the studies were lower in women. So just like to give you some perspective to think about, it depends on the health of the men and the women being tested. Um, it depends on kind of what their bodies are recovering from or maybe preparing for. So just keep that in mind. Patients are individuals. Okay. Androgens are hormones that actually stimulate the red blood cell production. So that's, you know, considerably more depending. Some women might have some during menstrual losses. In some women, it might be infinitesimal to undetectable, which might be more highly likely depending if there's menorrhagia involved. So, and menorrhagia is like extreme bleeding, which some people do experience. It's actually like a clinical level, like needs to see a doctor and be managed in some cases. So hematocrit inversely is proportional to body fat percentage. So in some studies, so that's going to be kind of dependent on what type of population you're looking at. So it's just kind of an anecdote right now. All right. We're going to get on to the next um, talk, part 7. We're going to talk a little bit about erythrocyte production, some more, just some more stats and generalities, and kind of move forward with that. But I wanted to leave it now since that's about the 10-minute mark. And yeah, that sounds pretty good. Nice, digestible little bit of information. Well, I hope you all are having a really great day, and I will see